Alright, so we're going to begin discussing the next chapter over cell physiology. Alright, so chapter 7. Alright, so membrane structure and transport mechanisms. Alright, so first, before we start talking about any type of transport mechanism, alright, we have to first discuss cell membrane and its composition. Alright, so all cells require that they have a membrane. Alright, so it doesn't matter if you're a little bacterium, alright, or if you're a plant cell, alright, or if you're a animal cell. Alright. A cell, to be a cell, has to have a cell membrane. Alright, so the cell membrane, besides kind of forming this outside boundary of the cell, it functions as a barrier. Alright, so it is selectively permeable. Alright, so permeable has to do with its ability to allow substances in and substances out, alright, of the cell. But the term selective means that it's very particular in what it allows to enter the cell and what it allows to leave. Okay, so think of the cell membrane as a let's say a fence or a gate or a barricade, and there are different ways that materials can enter and leave a cell, but there are checkpoints in place. Alright, so the ability for the cell membrane to be selectively permeable is a reflection of the composition of the cell membrane itself. Alright, so looking at the structure of the cell membrane, if you will recall, the cell membrane is composed of phospholipids. Alright, so your phospholipids are amphipathic molecules. Alright, so if you recall what that term means, it means that in the same molecule, one part of the molecule is hydrophilic and the other part is hydrophobic. Alright, so in this case, the cell membrane is a bilayer of phospholipids. Alright, so you have two layers of phospholipids that make up this boundary, or this barrier between the outside environment and the internal cytoplasm. Alright, so there's this model for what a cell membrane looks like. Alright, it's called the fluid mosaic model. Alright, so what this means, or the idea of this model, is that there are the lipids, the phospholipids, that are changing places with one another. Alright, it's fluid, it's dynamic. Alright, things don't stay where they are. Okay, lipids are constantly moving back and forth, and it's a mosaic because you have proteins that are embedded in the membrane. Okay, and if you look at it from the surface or from below, look up at it from below, you have these little fragments of proteins that are placed and dispersed randomly throughout the phospholipid bilayer. So it looks like a mosaic. Alright, so looking at this fluid mosaic model. So here you have your lipids. Alright, so your lipids are wiggling and waggling. Alright, so your lipids can actually move back and forth, and they can actually even swap sides. Okay, and your proteins can move throughout this bilayer. Now, recall what the phospholipid molecule looks like. All right, so here you have your phosphate group. All right, and down below here you have your fatty acid tails. All right, so this part of the molecule is polar. This part of the molecule is nonpolar. All right, so hence the molecule is amphipathic. All right, so this is kind of a short, brief clip of the cell membrane. Now note what's taking place. All right, you have your lipid molecules here. All right, so you have your phosphate groups here, and in green you have your lipids. Okay, your fatty acid tails. And here you have in blue you have a protein. All right, so this is a transmembrane protein, and you have molecules of water that are entering and leaving the cell. All right, so notice how they're moving, how the bilayer is moving back and forth. It wiggles, it waggles. It's dynamic. It changes. All right, so the cell membrane is fluid. It has a fluidic nature to it. All right, so before this fluid mosaic model came about, all right, there was an older model called the sandwich model. All right, so the sandwich model was this idea that your lipids are basically your meat and your cheese, and then the proteins are basically the bread slices. All right, and your proteins are found on the exterior, and your phospholipids are found on the interior. And we know this isn't the case. All right, we know that you have proteins that are found on the periphery. All right, and we know we have proteins that cross the entire membrane structure. All right, so these proteins that are found on the periphery, either on the outside or the inside of the cell membrane. All right, so here or here, for instance. All right, they're not embedded in the membrane. They're attached to the outer surface. Okay, they're referred to as peripheral membrane proteins. These proteins that span across the membrane, so transmembrane proteins, are referred to as integral proteins. All right, so your proteins are attached to the surface and span throughout it. Okay. So looking at the composition of the membrane, right, from the perspective of the fatty acids, all right, they're involved. All right, so if you recall, all right, from the lipid lecture, all right, that fatty acids, all right, they can either be saturated or unsaturated, right? Well, your unsaturated fatty acids, they have double bonds, they have kinks, they bend, right? So, in other words, your unsaturated fats tend to be liquid at room temperature, right? So they're fluidic, all right? So if a membrane, cell membrane, is composed mainly of unsaturated fatty acids, alright, they're going to have a high permeability, a high fluidity. Alright, so permeability implies this idea that things can move between the fatty acids. Alright, because there are gaps. It's more permeable, it's more porous. Alright, it's fluidic because the fatty acids themselves, the phospholipids, are not as compact with one another. Alright, they're spaced. Now, saturated fats tend to be solid at room temperature, right? That's because they have the carbon atoms are completely saturated with hydrogens. Alright, so in this case you have straight chains, alright, no bends. Alright, so you have low permeability. Alright, so it's much more difficult for things to move between your phospholipids. Alright, because there's less space, fewer gaps, because your phospholipids are compact and close together, because these fatty acid tails are like layers. Alright, they can be stacked one on top of another. Alright, so the fluidity is lower too, because everything's more highly compact. It's less fluid. Now, in animal cell membranes, there's another component that you find in the membrane besides the phospholipids and proteins. You also have cholesterol. Alright, so cholesterol, again, is an example of a lipid. But in this case, this particular lipid is a steroid. Alright, so your steroids are also amphipathic. Alright, in that one portion of the molecule is polar, and the other portion is nonpolar. Alright, so cholesterol. Alright, the cholesterol helps to maintain the fluidity of the membrane. Alright, so as temperatures cool down, as they lower, the membrane fluidity is maintained. Alright, the permeability is maintained as temperatures cool. Alright, because as temperatures cool, molecules have less molecular motion and they become more rigid. Alright, so the cholesterol increases the amount of space between your phospholipids. Alright, so you maintain this fluidity at lower temperatures. Alright, even at higher temperatures, alright, as you increase the temperature, alright, you get increased fluidity because you have increased molecular motion. Alright, so the cholesterol molecules help to stabilize the membrane, alright, to keep it from being too fluid. Alright, so let's look at the effect that cholesterol has on the membrane. Alright, so you have these scientists, and they constructed little bitty lipid bilayer vesicles. Alright, and then they embedded in this bilayer a certain quantity of cholesterol. And they wanted to see the effect that this has on the permeability of that bilayer. Alright, so they're looking at the movement of cholesterol. Alright, so based on their graph, alright, you have on the y-axis, you have the permeability of the membrane to cholesterol. On the x-axis, you have temperatures. Alright, so going from 0 to 10 to 20 to 30. Alright, so if you have 0% cholesterol, the membrane has the highest permeability. Alright, it's more fluid. Especially as you increase the temperature, it becomes more fluidic. Notice that as you as you increase the amount of cholesterol from 0 to 20 to 50%, all right, the permeability of the membrane to cholesterol decreases. Even as you increase the temperature, the permeability is lower than that without cholesterol. All right, so cholesterol has an effect on the permeability of the membrane. All right, so it helps stabilize it. All right, so if you recall, all right, this is an image of the endomembrane system. Okay. All right, so the endomembrane system has to do with proteins that are going to be secreted or have some other function elsewhere in the cell. All right, so you have your proteins going from the rough ER here. They're transported through a vesicle to the Golgi body. They make their way through the Golgi body where they're packaged and sorted, intact where they need to go, and then they get exported. All right, so some proteins get exported from a cell. Other proteins and structures get embedded into the membrane of the cell. All right, and this is all through the endomembrane system. All right, so looking at these proteins that are embedded in the membrane. All right, so these integral proteins. All right, the amino acids that are going to be interacting with the polar region of your bilayer. All right, so your phosphate head groups are going to contain polar or charged amino acids. Those that are found inside the bilayer, all right, associated with the bilayer, are going to be your nonpolar amino acids. All right, so polar outside, nonpolar inside. All right, so the nonpolars are the ones that are interacting with the lipids out here. Now, that means on the inside of this protein, in this region, you actually have your polar and charged amino acids on the inside, and your nonpolar sticking out on the outside, interacting with your fatty acid tails. All right, so looking at the function of proteins. All right, so this is kind of a review from the previous chapter over proteins and their function. All right, so a lot of this is going to be the same. All right, so proteins are used for transport. all right, which we'll talk about in a little bit. Proteins, most proteins are enzymes that speed up chemical reactions. You have proteins that act as receptors that bind to external signaling molecules. You have cell-to-cell interactions and recognition. And you have proteins that make up the cytoskeleton and extracellular matrix of cells. Alright, so proteins have other functions than just being enzymes. Alright, they're involved in transport, they have structural roles, they have receptor roles, they have communication roles. Alright, so they're pretty varied. This kind of gives you a big picture view of an animal cell membrane, all right, including the extracellular matrix. All right, so the big picture and what it looks like. Okay, so now we're going to start talking about transport mechanisms. Alright, so here you have a bilayer. And this bilayer is suspended between glass, separating two parts of the container with water. Now let's say that on one side of the container you add, let's say, salt. Or better yet, not use salt, let's use glucose. Alright, so all these little particles are glucose. Alright. So if this is a bilayer, what is going to affect the ability for glucose to go from this side of the container to this side? Well, it has to do with the fatty acid composition. Alright, so whether you have saturated or unsaturated fatty acids in the bilayer, that will affect the permeability. Whether you have cholesterol present or absent, whether or not you have proteins embedded in the membrane that aid in transport of certain molecules, whether or not your fatty acid tails are short or long will affect it as well. Alright, so there are a lot of variables that can affect the permeability of the membrane. Alright, so the cell membrane being a barrier. Alright, so this is a great illustration. Alright, because on the outside of the cell, you have a particle that's trying to enter. And on the inside, you have a particle that's trying to exit, but it can't. It's blocked. Alright, so this is showing you that the cell membrane is selectively permeable. Alright, it allows some things to enter and leave, and others is going to regulate, highly regulate, whether it can enter or leave the cell.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Alright, so here, looking at the permeability of the cell membrane. Alright, so small nonpolar molecules. Alright, so things like oxygen gas, CO2 gas, nitrogen gas, carbon monoxide gas. Alright, these things are really, really small. Alright, so they have a high degree of permeability. Alright, so that means they can slip in between the fatty acid molecules, slip in between the phospholipids, and enter a cell, and also leave a cell. Alright, so your gases are freely permeable. Okay? Now, small, uncharged polar molecules. Alright? They're not as permeable as gases, but they can still enter and leave the cell relatively easily. Molecules that are larger, like glucose and sucrose, that are also polar, like your some of your amino acids, for instance, right? They are going to have more of a difficulty entering and leaving the cell, right? And then lastly, the least permeable substances are going to be your ions. Alright, so your cations and your anions. Alright, so anything that's charged is going to have the least degree of permeability.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Alright, so the first transport mechanism we're going to discuss is the process of diffusion.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Alright, so here in this little animation, you're being shown the process of simple diffusion. Alright, so initially you had the movement of these little purple spheres, alright, that were over here. And they're moving to the right, alright, where they're least concentrated. Alright, so initially there's a net movement in that direction. Alright, so in simple diffusion, particles move from an area of high concentration to low concentration. Alright, so initially there is a net directional movement from high to low. Okay. Now over time, these particular particles will evenly spread out so that there's an equal proportion of these particles on both sides of the cell. Alright, so you'll have an equal or fairly equal concentration. Alright, so over time you have lower or at least a decreased net flow of particles until there's no net direction of movement of particles at all. Alright, so even at what we call equilibrium, alright, so at equilibrium you have equal concentration of particles on both sides of the membrane, you still have movement of particles across the membrane. Alright, so simple diffusion. Simple diffusion is a passive transport mechanism. Alright, passive transport mechanisms involve the movement of substances down their concentration gradient. So you're always going to go from high to low concentration. This process requires no expenditure of energy by the cell. Alright, so it doesn't cost the cell any ATP. Alright, the energy itself is the concentration gradient. Alright, so that's what's driving the movement of the particles. Alright, because this is a completely random process. Molecules themselves vibrate. These particles are bouncing off one another, colliding. The collisions are forcing the particles in one direction or another. So it makes sense that over time, if you have a whole lot of particles over here, they're colliding with one another. And so more of those particles are going to move in this direction. But over time, you're going to have collisions on this side where particles begin to move over here. Until eventually, everything evens out and you're at equilibrium, where particles are moving at an equal rate in both directions. Alright, so that's what's being shown here. You have a bilayer. You have a solute particle on this side, and another type of solute particle on this side. And the bilayer is freely permeable to both solutes. Alright, so that means initially solute A moves to the right. And it means that solute B moves to the left. Alright, because on the left side you have more particles of A that collide with one another and they go towards the other side of the container. On the right side you have more particles of B, they collide with one another and move to the left side of the container. Alright, and this is going to go back and forth until eventually you have an equal proportion of particles on both sides. You reach an equilibrium.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
Alright, so the diffusion of water is referred to as osmosis. Alright, so here you're looking at the availability of free water molecules. Alright, so notice here in this little animation, you initially have one side of the container with a high concentration of water and very little solute, or sugar in this case. And on the right side of the container, you have a high concentration of sugars initially and a lower concentration of water. Well, why is that the case? Why is it that you have a lower concentration of water on the right side initially? Well, think about it. Let's say you have two containers. One container, you add pebbles into. And the other one, you don't have any pebbles. Alright, and then you add water. When you bring this up to a volume of 10 mils, and then you bring this up to a volume of 10 mils. Alright, which container has more water is available? This one. Alright, the same thing's going on in this little animation. It has to do with the free water molecules. Alright, so remember what happens when you add a solute that's soluble in water to water. You're going to have water molecules that surround that solute particle. Alright, so if this is like glucose, right, for instance, a molecule of glucose, alright, you're going to have water molecules that are going to form this little hydration sphere surrounding and encasing that solute particle. Alright, so that means that that water is bound to something. Alright, it's not free. It's attached to a solute particle. Alright, so on the right side of the container, you have a higher concentration of solute initially. Alright, so diffusion dictates that substances move from an area of high concentration to low concentration. Alright, so water is going to move down its concentration gradient, where it's most concentrated, here, to where it's least concentrated, here. So, in this case, there's a net movement of water to the right. And so the volume on the right side of the container increases, and the volume on the left side decreases, until there is an equal concentration of water molecules on both sides of the container. Alright, in this example, the membrane is permeable to water, but impermeable to the solute. Alright, so the same thing's taking place here as in that little previous animation. What I like about this is it's showing you the water molecules surrounding the solute particles. Alright, so on the right side of the container, you have more solutes. So more water molecules are bound to solute particles. So there's a lower concentration of free water on the right side and a higher concentration of free water on the left side. Alright, so you're going to have a net movement of water left to right. And this is going to take place until the concentration of free water molecules is fairly equivalent on both sides of the membrane. Alright, same thing here. Higher concentration on the right side, lower concentration on the left, a net movement left to right until there's an equal concentration on both sides. Alright, even at equilibrium, water is still moving across this membrane, back and forth. Alright, it's not a static process. It's just that at this point, it has reached equilibrium. There's no longer any net diffusion of water across the membrane. Alright, so let's look at some applications of osmosis. Alright, so tonicity has to do with the ability of a cell to take up and lose water. Alright, so there are three key terms you need to become familiar with: isotonic, hypertonic, and hypotonic. Alright, so let's start with one of the easier terms. Alright, isotonic. Alright, if a solution is isotonic to a cell. Alright, so let's say you take a cell and you place it in an isotonic solution. That means that it contains an equal concentration of solute particles to those that are found on the inside of the cell. Alright, so what that means is, in an isotonic solution, water is going to enter and leave the cell, but there is no net directional movement of water. The volume of that cell is going to pretty much stay the same if it's immersed in an isotonic solution. So equal tonicities. Alright, that's the word iso is referring to. The same. Alright, so let's say that the solution outside the cell has a lower concentration of solutes. Well, in this case, you have immersed a cell in a hypotonic solution. That means that the inside of the cell is hypertonic. It contains a higher concentration of solute. Alright, so that means that water is going to move into the cell. It's going to go from area of high concentration of water to the area of low concentration of water. Alright, so in this case, if the cell has no way of regulating its solute concentration inside the cell, it has no way of expelling excess water, the cell will lyse. It'll burst. Alright, so what if you place a cell in a hypertonic solution? Alright, that means that that solution has a higher concentration of solutes than inside the cell. Alright, so that means that water is going to move from an area of high concentration to low concentration. In this case, it's going to move outside the cell because the inside of the cell is hypotonic. Alright, so as a result, the cell is losing water to its environment. It's shriveling up. Alright, so let's look at what happens to animal cells and plant cells when they're placed in these respective solutions. Alright, so looking at an isotonic solution. Alright, so an animal cell placed in an isotonic solution maintains the same volume. Right? And maintains a proper shape. Alright, water is entering and leaving the cell, but there's no net movement. Alright, so the fluid that surrounds our cells, the extracellular fluid, the fluid that's in our vasculature, is isotonic to our cells. Okay? Now, if you place a plant cell in an isotonic solution, it's going to lose some water to the outside environment. Alright? It's going to lose a little bit more than it's taking in. Alright? So it's going to become flaccid. It's going to become limp. Alright, so it's kind of like, lettuce that's wilted. Alright, if you place it in cold water, it rehydrates. Alright? Basically, it's losing water to its environment, even in an isotonic solution. Alright? It's going to lose a little bit of water, so it's going to become limp. Now, if you place an animal cell in a hypertonic solution, it's going to lose water to the outside environment. Alright, so that red blood cell will shrivel, for example. We call this process of shriveling crenation. Alright. If you place a plant cell in a hypertonic solution, it becomes plasmolyzed. Alright, so notice what's taking place here. Your central vacuole has shriveled. Alright? And your central vacuole in a plant cell usually takes up a majority of the volume of the cell. But now all of your internal structures are found in the center. And your plasma membrane has actually pulled away from the cell wall. Alright, so the cell has lost water. Alright, to its environment. Alright, so this is very unhealthy for a plant cell, as well as an animal cell. Now, what if you place an animal cell in a hypotonic solution? Well, in this case, water's entering the cell because the internal constituents of the cell are more concentrated, and so it's going to burst. Alright, so recall that animal cells don't have a cell wall. Alright, so they can't maintain their shape and structure without having some mechanism to remove this excess water. Alright. Now, plant cells placed in a hypotonic solution, they're going to become turgid. Alright, they're going to swell up. Alright, notice that the central vacuole has swollen, takes up a majority of the volume of the cell. Alright, all of your organelles get smushed along the edge of the cell membrane. Alright, so here you're going to look at the process of plasmolysis. Alright, so your cells are going to lose water to the environment. Alright, so notice what happens to the animal cells, the squares. Notice what's happening to the chloroplasts. They're kind of moving to the center of the cell instead of being at the periphery. Alright, the cell size is shrinking. The cell membrane is pulling away from the cell wall. Alright, so let's look at the opposite. Alright, so in this case, putting a plant cell in a hypotonic solution, that plant cell is going to swell up and become rigid, turgid. Alright, so look at the plant cell. So initially your little chloroplasts are going through cytoplasmic streaming, moving around the periphery of the cell, and now your cells are beginning to swell up and enlarge. That central vacuole is becoming much, much larger. As everything gets smushed on the edge of the periphery of the cell. Alright, so the next topic we're going to discuss is a process called facilitated diffusion. Alright, so facilitated diffusion uses proteins that aid in the movement of molecules across the membrane. Alright, so water is small and polar. Alright, so it can diffuse through the cell membrane, but it can't diffuse fast enough, alright, for organisms to really adapt to their surroundings in order to survive. Alright, so there are proteins called aquaporins that aid in the movement of water across the membrane. Alright, so here you're being shown the aquaporin protein and the movement of water molecules through this protein. Alright, so here is a cartoon image figure of of this aquaporin protein. Alright, so here this is showing you that water is able to diffuse out of and into the cell by itself. But it needs a little help. Alright, so you have this protein, aquaporin, that allows the movement of water into and out of the cell much more readily. Alright, so it occurs at a faster rate. Alright, so this aquaporin protein only transports water. Alright, so talking about facilitated diffusion. Facilitated diffusion is another example of passive transport. Alright, so again, passive transport. Molecules, solutes, move down their concentration gradient. Okay? And it requires no expenditure of energy by the cell. So no ATP has to be utilized. Alright, facilitated diffusion is diffusion, but in this case, these solute particles require the use of some type of integral protein to allow the movement of particles across the cell membrane. Okay, so it's facilitated. It's helped. Alright, so it's kind of like walking an elderly lady across the street. Alright, for example. Okay, that's what these proteins are doing. They're helping the movement of things across the membrane. Alright, so you have two different types of membrane proteins that are involved in transport. Alright, so you have channel proteins. Alright, so think of a channel protein as a pipe. Alright. So basically, the solute particles just zip right through the pipe. Alright, so here you have solute particles moving through this protein from inside the cell to outside. Okay. A carrier protein is a little different. Alright, it's more like a Pac-Man. Alright, so here your carrier protein opens up on one side of the cell. It allows the binding of the solute to a receptor site or binding site in the protein. Once bound, that transport protein opens up to the other side. Alright, so it's a little Pac-Man. It opens and closes and allows the movement of the solute particle. Now, your transmembrane proteins that are involved in facilitated diffusion are very specific. They transport very specific solute particles across the membrane. Alright, so here you have the movement of, for instance, glucose into the cell. Alright, so glucose being a polar molecule, it's a little bit larger. It requires a protein molecule to enter the cell. Alright, so glucose can enter by binding to a membrane protein. Alright. So you have this little binding site, this little pocket that's going to accept glucose. Now, once the glucose molecule binds, notice there is a conformational change in the protein. Alright? This end of the molecule closes in on itself. Alright, the mouth closes. And now the other end of it opens up. And once it opens up, the glucose molecule can leave. And once it leaves, it's no longer bound, it goes back to this original configuration. Alright, so the proteins are facilitating the movement of the particles across the membrane. Alright, so this is also how ions get across the membrane. It involves membrane proteins. Alright, so looking at active transport. Alright, so active transport. In this case, you're moving solute particles from an area of low concentration to an area of high concentration. Alright, so you're going against the concentration gradient. Okay? So, in active transport, think of passive versus active transport in this manner. Alright, so let's say that you and some buddies go on a canoe trip. Alright? And let's say that you're going to go downstream. Alright, so if you're going downstream, the flow of the current is going to be enough to drag you downstream. Alright, so in this case you're going high, alright, to low. Alright, so this is passive transport. Alright, so you can just kind of lay back, chill out, and you're going to go downstream. Not a problem. And you don't have to even exert any energy. Alright? So, no energy input. Okay? Now, active transport. Active transport, alright, you're going to be going upstream. Alright, so you and some buddies want to go upstream. Alright, in your canoes. You're adventurous. Alright, so you're going to go from low to high. Alright? The currents are going to become much more torrentious and challenging as you go upstream. You're going to have to paddle and use energy to move upstream. Alright, so you're going against the flow, the natural flow of the water. So you're going against the concentration gradient. Alright, and you're having to use energy. Well, active transport, the cell has to use ATP, or energy, to move solute particles against their concentration gradient. Alright, so a classical example of active transport is the sodium-potassium pump, alright, which we'll get to in a minute. Alright, so this is a short animation on the sodium-potassium pump as an example of active transport processes.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Alright, so this kind of illustrates the sodium-potassium pump as an animation. Alright, so your pump, or your integral protein, has 3 binding sites for sodium ions. Alright, so when they bind, you have a ATP molecule that undergoes a hydrolysis reaction, which allows the cleavage of one of your phosphate groups, and it's attached to the protein. Alright, so you're transferring energy. Alright, so this is a phosphorylation reaction. Alright, so that protein becomes phosphorylated. Alright, so now once phosphorylated, this part of the protein closes and the opposite end of the molecule opens up. Now, the sodium ions can leave. The potassium ions can bind to their respective binding sites. The phosphate attached to the protein is removed through a dephosphorylation reaction. This end of the molecule closes, the other end opens up, and potassium enters the cell. Alright, so the sodium-potassium pump. Sodium is moving against its concentration gradient, and potassium is moving against its concentration gradient. Alright, so in this case, both ions are moving against their concentration gradients. Alright, so when you're talking about a cell, alright, the membrane of a cell is charged. Alright, the outside of a cell has a net positive charge. The inside of the cell has a net negative charge. Alright, so you have a higher concentration of cations outside and a higher concentration of anions inside. Alright, in this case, you're setting up what's called a membrane potential, or a voltage across the membrane. Alright, so talking about this membrane potential, this voltage across the membrane, how exactly does a cell maintain this voltage? Alright, well, in this case, you saw that the outside of the cell has a positive charge, and the inside has a negative charge. Alright, net charges. Okay. So, what the cell establishes is something called an electrochemical gradient. Alright, so let's break down this word, electro and chemical. Alright, so in this case, you're talking about ions. Alright, so hydrogen ions. These hydrogen ions are pumped outside of the cell. Alright, so they're pumped against their concentration gradient, which requires the use of ATP. Alright, so you build up a chemical gradient of protons higher outside and low inside. So you get a chemical gradient, but then you also have an electrical gradient. Alright, a voltage. Alright, so as a result, you end up with a positive charge on the outside and a negative charge on the inside. Now, cells can couple. A process that involves active transport with one that involves facilitated diffusion. Okay. So, in this case here, alright, we're looking at coupled transport, where the cell is pumping out protons into the extracellular environment against its concentration gradient. Requires ATP. Alright, so these protons are then high outside and low inside. Alright, so you have higher protons outside. So you have this co-transporter protein. That's a sucrose proton co-transporter. Alright, so for the cell to move sucrose into the cell, you also have to have the binding of protons to this transporter. And so both of these molecules move down their concentration gradient through facilitated diffusion. Facilitated diffusion. Alright, so let's kind of think about this process here. Now, what would happen if you were to place a cell in an aqueous environment that's increasingly acidic? Would you expect the rate of diffusion of sucrose entering the cell to be faster or slower? Alright, well, if you're pumping protons out into the outside environment, outside the cell, alright, so you're lowering the pH. Alright, so if you lower it even more, you would increase this process of pumping sucrose in. Now, what if you were to increase the pH? Alright, so you're becoming more alkaline. This would actually slow down the process of pumping sucrose into the cell. Alright, so this kind of gives you a review of passive and active transport. Alright, so looking at diffusion, facilitated fusion, active transport. Alright, and it's a nice little summary slide. Alright, so the one thing it does leave out is osmosis. Alright. Osmosis is also another type of passive transport. Alright, so now we're going to move on into talking about bulk transport. Alright, so exo and endocytosis. Alright, so exocytosis, you're going to be releasing molecules in vesicles outside of the cell. Alright, it's proteins, carbohydrates mainly. Endocytosis, you're bringing things into the cell. Alright, so here, as you can see in this little short animation, you have a vesicle that has some type of molecule that's going to be secreted outside the cell. Alright, so this is going from the Golgi apparatus to the membrane. Alright, so the vesicle, the membrane of the vesicle fuses with the membrane of the host cell. The contents of the vesicle are then expelled outside the cell. Alright, so this is exocytosis.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract. Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
Alright, so here you have your little vesicle. Alright, so inside the vesicle, you have your own little microenvironment. Alright, so you have this little bilayer that makes up the membrane of the vesicle, and it can contain proteins, carbohydrates, things that need to be transported outside the cell. It could be lipids, so potentially hormones, for instance, would be another example of something that could be secreted from the cell. Alright, so here you have the vesicle, the membrane of the vesicle that's actually fusing with the cell membrane. The contents of the vesicle are then expelled outside of the cell.
In endocytosis, membranes invaginate, or pinch in, to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
Alright, so here you have a white blood cell that's engulfing this red particle here. Alright, so it gets trapped inside of the cell. You can see it kind of wraps around and encases it, envelops it. Alright, so that's the process of phagocytosis or cell eating. Alright, so here you have a alteration in the structure of the cytoskeleton. Alright, allowing for the membrane to expand out. Alright, so you have actin filaments that are rearranging so the cytoskeleton can alter itself to wrap around this food particle. It's then engulfed, encases it, and forms a vacuole. That vacuole, once it enters the cell, will then fuse the lysosome, forming a phagolysosome, where the contents will be digested and released into the cytosol. Alright, so here you're being shown an image of a white blood cell that is engulfing a bacterium.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells in capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
Alright, so this is basically a random sampling of the fluid and its materials outside the cell. Alright, these materials are brought in as the membrane invaginates in on itself. Alright, and then once inside the cell, those contents are released.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
Alright, so here, this is similar to pinocytosis, except for specific molecules brought into the cell. Alright, so you're not taking a random sampling. Alright, so you have molecules that bind to specific receptors on the host cell membrane. When they bind, those contents are brought into the cell.